Hello everyone, I'm Jasmine and I'm the one with the pressure of going last. So here I'm going to talk to you about my research I've done with Dr. Ban on protective antigen and its mechanism in pore formation. So this is biochemistry. There will be a lot of science terms, so be prepared. First I'm going to go an overview over what I'm going to be talking about today. And I'm going to start off with the background of anthrax and how it works. I'm going to get into the precise mechanism of anthrax, exactly what components make it form a pore, and a mutant that we worked on, the D425A protein. And then I will continue with my research, how I started with an Alexa Fluoro 350 label, and then I went on to do research on an unlabeled protein. And I will finish off with the implications of my research and acknowledge those who helped me. All right, so anthrax is a disease caused by the bacterium Bacillus anthracis, and this is a two-component system. So it's the interaction of two moieties, protective antigen, and enzymic moieties, either lethal factor or edema factor. It is the interaction of protective antigen with one of these enzymic moieties that leads to the toxicity of anthrax. Specifically, how this works. So we have protective antigen, and it binds to a surface cell receptor, and then that allows it to be cleaved off. Part of protective antigen is cleaved off. This will then allow it to form a pore, and this pore acts as an anchor for one of the enzymic moieties, either lethal factor or edema factor. Then this will be endocytosed onto its host, endosome, and then under acidic conditions, one of these enzymic moieties will release its toxin onto its host. So here we have cryo images of the protein. So here we start off with a non-toxic heptamer of protective antigen. But when it releases its toxins onto its host, it ends up as a pore. The mechanism of how it goes from a non-toxic heptamer to a pore is not well understood, and that is what we study in the lab. In order to study this mechanism, we decided to study a protein that was not able to form a pore, and we came up with the D425A mutant. Collier did research on this mutant, and he found that it was not capable of forming the pore. So I have here a CDS page. And what is important that we have 3 proteins, and 2 of them are mutant proteins that have an aniline mutation. So under acidic conditions, you're able to form a pore. But here we have that the wild-type protein is capable of forming the pore in acidic conditions, but these 2 mutant proteins are not able to form the pore. So for this reason, we decided to use this mutant protein for our study. He also did research and found that this mutant could form a heptamer and could bind to one of the enzymic moieties' lethal factor, but it could not express or translocate the toxin. There was also cryoimaging data on the mutant protein, but the cryoimaging data is only top views. There had not been any side views of the mutant protein. So what we found is an intermediate structure of the mutant protein. To do this, we started off by labeling both the wild-type protein and the mutant protein with the Alexa 350 label. Then we went on to do fluorescence and ended up analyzing kinetic data of our proteins. We also collaborated with Case Western University to do cryoimaging studies on the protein. As I mentioned, we started off with labeling the protein. I use an Alexa 350 label, and that allows us to measure the fluorescence at an 18 angstrom distance from a tryptophan. The mutation of the protein is right there in D425A. So it's a cysteine that is changed to an aniline. So in order to understand the precise mechanism of pore formation, we have a protocol. So we start off by isolating our mutant protein. Then we go on to do fluorescence and kinetics on our protein. We compare the results of both our proteins, the wild type and the D425A mutant. And in the end, we hope that this serves as a better model for understanding pore formation. What I have found with my previous studies with the labeled protein is that, so here we have fluorescence, and we have that the wild type protein on top and the mutant protein in the bottom. And the wild-type protein has a change in fluorescence under acidic conditions, which is expected. And the D425A mutant also experiences some sort of change in fluorescence, which was a bit to a surprise because it's not able to form a pore, but it does experience similar fluorescence change to that of the wild-type protein. We decided to do kinetics on the protein, and the wild-type protein has an initial structure change and then continues with some sort of other structure change. The mutant protein has an initial structure change, but then it stops. So that means that there's a stop in pore formation. This is also to a surprise because we didn't expect the mutant protein to experience any sort of structure change. In order to quantify to see if the mutant protein was experiencing similar rate of formation to that of the wild-type protein, we decided to check the activation energy of the proteins, and they were similar. So the activation energies of both our proteins are comparable. So this made us think, is this due to the dependence on the mutation or maybe to the label that we had on our protein? So we decided to do the studies without labeling the protein. So here I have the kinetics of the wild-type unlabeled protein. And again, we see a structure change, but then it continues to form a pore. Now with the mutant protein, we see a structure change again, but again, a stop in pore formation. So this confirms our previous studies. Now we collaborated with Case Western University, and they helped us get cryo images of our D425A mutant. And here in the yellow boxes, I have that outside of the heptamer, there is electron density, indicating that this could be the intermediate structure we hypothesize that the D425A mutant experiences. So we revised our hypothesis. We used to believe that the pore formation was a multi-step mechanism that went from a heptamer to a pore. But now we revised our hypothesis to include an intermediate structure. So these new studies correlate with our previous studies, and the intermediate structure that we have found is on pathway to the wild type. And in the future, we will plan to redo the studies, maybe with a different mutant, or perhaps a different label that measures the fluorescence at a smaller wavelength. And I want to thank everyone in lab who helped me, and also our collaborators, and the funding that we received.